Et nous avons le grand plaisir aujourd'hui d'accueillir Maria Chait, qui est venue d'un pays lointain, l'Angleterre. N'est pas encore totalement séparée de nous, vous voyez, sur le plan des interactions intellectuelles. Donc nous avons beaucoup de chance aujourd'hui. J'espère que ça durera. Et I switch to English to welcome Professor Maria Chait, who is from University College London. She's heading a group inside the Ear Institute, l'Institut de l'Audition. This is, as I understand, an institute similar to what Christine Petit is launching here, dedicated to understanding the ear, but also the brain behind the ear and how the analysis of the auditory signal progresses through the brain. So she's also a member of the Institute of Cognitive Neuroscience, or affiliate of the Institute of Cognitive Neuroscience in Queen Square in London. This major institute has been making dramatic progress in the past 30 years on brain imaging. So welcome, Maria, and thank you for telling us about how the brain discovers patterns in sound sequences.
Thank you. It's a pleasure to be here and share my work with you. My lab at the Ear Institute is using a variety of approaches, including behavioral and brain imaging approaches to understand auditory processing. This is our recent anti-Brexit protest designed to show that our lab, although in England, but is very international and will be, like all research in the UK, will be severely affected with this unfortunate outcome. In the lab, the question that we're interested in addressing is related to what I tried to depict in this picture. So we're interested in understanding how listeners use sound to understand their surroundings. So normally when we think of the auditory system, we automatically think of speech. But in fact, if you think of it, speech is a relatively recent development. In fact, our brain has evolved, our auditory system has evolved perhaps for something different altogether, and that is to facilitate survival in the environment. Indeed, our ears are open all the time, unlike our eyes. And it's hypothesized, and this hypothesis is at the basis of most of what we do in the lab, that the auditory system serves as an early warning system. So it continuously scans the environment for potentially important events and then brings them to your attention. So it's very common, it's a very intuitive understanding that we very often hear important events and then we look at them. And it's this aspect of auditory processing that I'm interested in studying. And specifically, the kind of questions that we're interested in the lab involve understanding how listeners make sense of their environments using sound, and what information from sounds listeners care about in this process, and what aspects of auditory processing take place automatically, and which ones require attention. And this has, in addition to the basic research, there's a variety of applications for this knowledge. So in understanding complex forms of auditory deficits, but also for designing alarm systems, hearing aids, and so on. And the challenges in this kind of work, and this is how work in the lab is constructed, are to design simple sounds with which we can model and study these complex auditory processes, and then understand the brain correlates of these processes, so understand with EEG or MEG what the brain does. And today I will talk about one particular aspect of this work that is related to understanding sensitivity to patterns. Sensitivity to patterns is a feature of all sensory systems. In hearing, much of what we know from sensitivity to patterns comes from this field of study that's called the mismatch negativity. So the mismatch negativity is a brain response that is generated when some sequence is violated. So you can create a pattern as complex as you want, you can violate it, and then measure the response to this violation, and that is used as a signature to indicate whether the brain was sensitive to this pattern or not. I'll talk about this more later. But the outcomes of this work suggest that the auditory system is exquisitely sensitive to a variety of different patterns. And this makes sense from an etymological perspective, because of course many, or maybe most, of the sounds that are relevant to us, that we want our brains to detect, are statistically regular in some sense. That's because it's likely that the sounds that we care about, that our brains care about, are generated by animate objects. And animate objects are sort of by definition constrained by the physics of the body. And hence the sounds that they produce are also constrained in different ways. So, and we use this information, sort of this information of pattern and sound, in order to first identify what a sound is. So this is an example. If I play just one sound, it's going to be very difficult to understand what it is. But if I play a whole sequence of them, it's immediately clear what the sound is. But in addition to recognizing the sound, you learn a lot about what's going on. You know that a person is walking, you can tell how quickly they're walking, where they're walking, perhaps something about their body, something about the surface that they're walking on. And all of this information is information that we derive from a pattern. I mean, in addition to that, detecting patterns facilitates listening. So when we heard the sound of footsteps, in addition to recognizing it, we were also able to immediately predict the next sound. So that's inherent to detecting a pattern. And that's important because it helps us it helps us process efficiently sounds that were expected. So in the case of footsteps, if I hear a footstep approaching me, then I will move. In the lab, we study this using simpler sequences like this, and it's been known for a while that it's easier to track sequences when they are regular relative to when they're random. So these kinds of sequences, so temporally random sequences, are harder to attend to than regular sequences like this. And this is measured by asking people to respond to a certain sound in the sequence, and they're usually much faster and more efficient when sequences are regular. And that is also true when you present them in a mixture. So what this shows is a situation where there are two sequences of sounds presented simultaneously. There's a random one and a regular one, and I'll play this in a second, and it's always easier to attend to the regular sequence than to the random sequence. It might be difficult in this big room, but you can give it a try. So try to listen to them and see which one is easier to track. So there's a high one and a low one. So the high one is regular. And the way these kinds of findings are usually explained is by saying that when you listen to a mixture like this, and when your brain discovers that one of the parts of the mixture is predictable, it's just easier to anticipate future events and then pull them out from the mixture. So it will be easier to focus your attention on a regular sound. And similarly, it's also easier to ignore regular sounds. So this is an illustration from a study that we ran recently, where we presented a similar mixture to what you heard before. There were two sounds. In this case, both of them are temporally random, and they're presented concurrently. Participants have to focus on a random sound, so they're always trying to track a random sound, and they're detecting some, so occasionally we have some special tones that they have to detect in the random sequence. And what we vary is the regularity of the ignored sound. So it could be either random, like here, so both of them are random, and it sounds like that. Try to see if you can track the high tone. Or the other thing that we can do is we can make the ignored tone regular. Still ask people to to track the random tone. So this sounds like that. Still try to focus your attention on the high tone. And what we find in these series of experiments, this is what's shown here. So these are the different conditions that we used. The values here are the status of the ignored tone. The attended one was always random. And the ignored tone can be either random or regular, with what these numbers indicate is rate. So this one was relatively fast, so there was 100 milliseconds between tones, and this one was relatively slow with 400 milliseconds. And what we find is that for moderate speed, you always do better when the ignored sound is regular, suggesting that it's easier to suppress it. And the explanation is similar. So in the same way, so you're trying to focus your attention on this one. This one is regular. Your brain is able to predict future events. And so it's easier to suppress it and focus attention on this one. Similarly, so this is one example where being able to predict what is going to come next will facilitate processing. Similarly, being able to predict what comes next, which is associated with the sensitivity or detection of a pattern, also enables us to detect change quickly. So if we go back to the footstep example, so if you hear somebody walking behind you and the footsteps suddenly stop, that can indicate a very important, potentially critical event. And if you have detected the pattern, so you can detect the end of the pattern very quickly as soon as the next event that you were expecting doesn't arrive. And we can measure that in the lab similarly, so we can use these regular patterns that we occasionally stop. And what we predict is that the speed with which listeners are able to detect the pattern will basically depend on the intertone interval. So if we stop the pattern here, the listeners will wait that much, discover that a tone is not coming, and respond. And indeed, that's what happens. So we are very, very efficient with detecting offsets, even with complex patterns, so much simpler than an isochronous pattern that I'm showing here. And even more, perhaps more remarkably, we can see evidence of this happening with stimuli like this that model much richer scenes. So what is shown here is a spectrogram. So this is time over here and frequency over here. And there are 1, 2, 3, 4, 5, 6, 7, 8 concurrent sounds happening at the same time. So this is our attempt at modeling a scene with many different sounds, like a party. Many different sounds present at the same time. What we do with this kind of scene, so it sounds like this. It's just a mess. And importantly, in this example, each sound is irregular, so temporally irregular. What we can do is we can introduce changes in these scenes, so just have one source appear in the middle and measure how quickly people are able to detect this. So this sounds like that. You heard the new one appear. Interestingly, so we can use scenes like this. We can also make them regular, like here, and also introduce a change. I don't have to play that, actually. And what we find across several studies is that it's always easier to detect a change. So easier meaning your performance is higher when the scenes are regular relative to when they are random. And this is true for detection performance, but also for how quickly you respond. So you respond faster when scenes are regular relative to when they are random. And the way to explain it is that in a scene like this, when you're listening to the onset and your brain discovers that everything is regular, you're able to predict future events, perhaps in each stream independently. And then when a new one actually comes, is really unpredictable. So you detect it very quickly. So this one sounds like that. So this onset of the new event pops out because it's unpredictable. Whereas in scenes like this, Because you cannot extract the structure from each of these sources, it would be much harder to detect a new event over here, despite the fact that it appears in a new frequency. It's just harder. And indeed, we can see evidence of this in the brain response as well. So this is data from MEG experiments where we record the brain responses of listeners who are listening to these kinds of sounds. And what you see here, 0 is when the change happens. So 0 is this point. And red is these changes that happen in random scenes. And blue is changes that happen in regular scenes. And you can see from the very first response to the change, so that happens just before 100 milliseconds after the onset of the change, you already respond, your brains already respond much stronger to changes that are presented in a regular context relative to changes that are presented in a random context, suggested that our brains extract this information quickly and use it to help you detect changes better. We can see these effects. So far, I've been showing you data where the pattern is a temporal pattern, but it doesn't necessarily have to be. So this is an example of a sound where the regularity is a spectral regularity. So temporally, there is a tone happening at each point in time. But here, the tones are arranged in a pattern. We can create new patterns on each trial. What we do is we introduce somewhere in the stimulus, we introduce a deviant tone. So it's this tone here. I can put a square around the pattern so that you can see that this one is a deviant. And that happens on some of the trials. And we ask our listeners to detect it. And this is what it sounds like. Try to listen out for the deviant tone. That was rather easy, right? So just to show you where it is, I can black out everything else. You can see that this one deviates and your brain is detecting it very rapidly. We can do the same thing. We can take the same tones, but we can now arrange them in a random order. This is what is shown here. We still have the same deviant tone over here. Yeah, so it's equally far from the frequency range, and this sounds like that. Did you hear the deviant? It was much harder, and it's indeed across participants really much harder. So they're both slower and worse at detecting these deviants that are presented in random contexts relative to deviants that are presented in regular contexts, despite the fact that we control for the long-term distribution and the exact alphabet sizes used. And the explanation for this would be very similar to the explanation that I offered before. When you have a regular pattern, you can predict upcoming tones. So in this case, you're predicting a tone over here, like in this pattern. When you get a deviant, when you get something that violates your prediction, you respond very quickly. But you are unable to do this as efficiently when the sequence is random. You can predict that a tone is going to come. You can predict that a tone within a certain range is going to come. But because the prediction is not as precise as a prediction in a regular case, you're also worse at detecting it. And indeed, we can measure this in the brains of naive listeners. So these are, again, listeners who are not performing the task now. We're measuring their EEG, and they're watching a movie. And we can see this response. So the blue traces are the responses to the deviants in the regular case. And the orange traces are responses to deviants in the random case. And you can see very early on, from before 100 milliseconds, there is an increased brain response to the deviants in the regular pattern. So in summary, this evidence that we have for sensitivity suggests, well, first of all, that we're sensitive to a variety of patterns. I've only showed you very simple examples here. And that we use these patterns to help us analyze the auditory scene and effectively interact with the auditory scene. And in general, this performance pattern is consistent with this theory of brain function that is called predictive coding. The idea is that we use information available to predict future events, and that helps us process the data process the stimulus more efficiently. But there is a remaining question, which in fact characterizes auditory research, but perhaps perception research more generally. We know that we're sensitive to patterns in all sensory modalities, but how do we detect them? How do we discover that a pattern is present? So as I mentioned before, in hearing, a lot of work that contributed to our understanding of pattern processing comes from this mismatch negativity literature. And that in itself is an indirect evidence to sensitivity to patterns. So we use we measure violation of patterns and use it indirectly to infer that we have detected a pattern. But what we're interested in is understanding how do we find patterns in the first instance. And this is the question that I'll talk about for the rest of this talk. So in the lab, our approach to trying to understand this started with a very simple stimulus, and it was led by a PhD student, Nicolas Barascoud, who is now in Paris, actually. We used random tone sequences similar to the ones that I've shown you before. So this is a spectrogram, this is time, and this is frequency. And we have a pool. So in all of these experiments, we have a pool of 20 frequencies that we will draw from in various ways. So in this example, I'm just randomly selecting frequencies from the pool and playing them. And the result is a sound like this. Just a random sequence of tones. What I hope you get out of it is that it's fast. Each tone is 50 milliseconds long. You can't really consciously follow it in any way. You can just let it flow through you and you know it's random. To understand what kind of information you extract from this signal, or how are you able to detect patterns in this kind of signal, what we can do is we can introduce a pattern partway through. So this is what is shown here. So this is a stimulus that starts off random, and then at some point, that's indicated with a line here, it changes into a regular pattern. I hope you can see it. The way we create the regular patterns, so in this case, it's a pattern that consists of 10 tones that we randomly select from the same pool and then repeat. And so that enables us to use a different pattern on each trial. And we can also change the complexity of the pattern by changing the number of tones we choose to repeat. So here it's 10. It's a relatively complex pattern. We can choose 2 tones to repeat. That will be a simple pattern, or even 1 tone. And we also change the time at which the pattern is present and whether it's present at all. And just ask our listeners to press a button as quickly as they can when they hear it. This is an example. So what I hope you get from this is that it's very easy to hear out a pattern. It doesn't require any effort, you just hear it. But in fact, it's too fast for you to reason, to sort of say, oh, I just heard this one repeat, and then that one repeated, oh yes, I can conclude that the pattern was present. It just pops out. So here's another example. Yeah. So it is easy, but it's not trivial. So if you think about what had to happen now in your brains when you were listening to this, you had to, so first of all, you can't detect the pattern based on the first cycle, right? Because the first cycle is just a random sequence of tones, not different from the random pattern. So you have to extract some kind of representation or some information from the random signal as you hear it, as your brain hears it. Your brain has to compare incoming tones to this representation and keep track of repetitions. And then at some point, when enough repetitions have been heard, it brings this to your attention, right? I mean, you didn't need to do it with particular effort. So we know that you can't do it during the first cycle. So you need to hear it repeating. But how many repetitions do you need? I mean, it's a non-trivial question because there's no single statistical measure that will tell you deterministically how many tones you would need. How much more information you need to detect that a pattern is repeating depends on the properties of the signal, but also on the settings of and limitations of your brain. So how much information does your brain need to conclude that the pattern is repeating. To try to give us a benchmark of what is statistically possible with this kind of signal, we used a model. I don't have much time to go into it in a lot of detail, but the idea is it's a statistical model that has infinite computational resources and infinite memory. Basically, for those who would understand, it's comprised of a nested chain of variable length Markov models. So it basically remembers all possible transition probabilities within the signal. And the model, when we run it through signals like this, so the model gives us an information content for each tone, which is on the y-axis. So tone is here. I've cut it, so imagine the random sequence continuing for a while. I've cut it to fit it in one slide. The idea is when the model is presented with random sequences, the tones are surprising because they're random, so the information content is high. When we transition to the regular sequence for a while, that doesn't change. The information content stays high. At some point, the model discovers that the statistics have been violated. So and that results in the tones becoming increasingly more predictable until the model has detected the pattern, and then the information value for each tone settles on a low level. So what we would consider as the model detecting the pattern is this point, the first point at which the information starts dropping. And this happens for the 10-tone cycles at a cycle, so 10 tones plus 4 tones. So the model doesn't need to hear the pattern repeating twice. It needs the first cycle plus 4 tones. And that's, in fact, a feature that's not dependent on cycle duration. So you can have it for cycles of 10, like I've shown you. But in fact, you will see it for any cycle. So for cycles of 5 tones, you need 9 tones, so 5 plus 4. For cycles of 10, this is what I've shown you, 10 plus 4. For cycles of 15, again, you need 15 plus 4, and so on. It's not important if you don't understand this bit. This is more for the aficionados, and the idea is that the model, the number of tones you need to detect the pattern should depend on the statistics of the random sequence. And the random sequence here is always the same. But back to the stimuli. So we have now an estimate of what is absolutely, the best, the fastest we can detect a pattern, which is a cycle plus 4 tones. This is the performance of the ideal observer. Now I want you to try to introspect, and I'll play a few examples again, how long it took you to detect the pattern. So I'll play it again. I know I shouldn't be taking questions at this point. I usually ask people to guess, but I will just ask you to introspect, and then I'll give you the answer. So in fact, people behave exactly like an ideal observer. So most people, most of you, in a properly controlled sound environment with headphones, would not need to hear the pattern repeat twice. You just need the first cycle and a few more tones to detect that it's repeating. And that's remarkable. And the way we measure it in the lab is we use these sequences, so transitions from random to regular. We also use the opposite transition from regular to random, which I will not talk about until the very end of this talk. And we also use these simple step stimuli as a control. So we ask people to detect these transitions as quickly as they can. And we use this stimulus to measure just how quickly they can respond to an obvious auditory change. And then we subtract the reaction time from this stimulus, from these two. And that gives us a sort of lower bound measure of how much information you need to detect the transitions. And the way the data look like is shown here. So overall, the size of the bar is the raw reaction time. And this dashed bar is the reaction time to the simple step change. So the orange bar is the reaction time for the transition stimuli in the random to regular case. And as you can see, they take a cycle and a half. So the first cycle you can't detect. They just need 5 more tones for cycles of 10 tones. And so like an ideal observer. Another way of looking at the same thing is looking at the distribution of reaction time across trials. And this is what's plotted there. And indeed, for the vast majority of the trials, people respond before it is actually repeated. And we can see the same thing for patterns of 20 tones, so much longer patterns. Everything is a bit more variable there, so there's larger error bars overall. But this property that you, on average, need a cycle and a half, and on most trials you respond before 2 cycles, is still maintained for these really long patterns as well. So this is interesting because these sounds, as you would, I'm sure, agree, are nothing like anything we experience in the environment. We would, I doubt our subjects who have done these experiments have ever had to hear patterns like this anywhere else. And the fact that they demonstrate this remarkable ability, you know, they basically perform like a statistical learning machine, is exciting because it means that with these simple sounds, we're probably tapping a system, an automatic system, where I'll show you an evidence for why we think it's automatic, that is probably useful for something else. You know, that the brain has probably developed to help us deal with sounds that we actually care about. And so it's interesting to understand the brain mechanisms that give rise to this performance. What we do to understand this is we use MEG. So MEG is a form of brain imaging. The picture is shown here. Participants are sitting. We measure their brain activity while they're performing a visual task. So in the brain imaging experiments, participants are always naive. They don't know that they're performing an auditory task. They think that they're there to do a visual experiment. And we tell them that the sounds are there to make it a bit harder and to prevent noises from outside of the booth where they're sitting to enter the booth. And the stimuli that we present are similar to the ones that I've shown you. So these are sequences with transitions from regular to random or random to regular. And half of the time there's a change, and half of the time there's no change. And I'll focus on these kinds of stimuli, on the transitions that we've talked about from random to regular. And at the very end, I'll say something about the other ones. So this, and another thing, MEG gives us, so we have 274 independent channels that we measure brain activity through. And this is an example of one condition. So each line here is one channel. To summarize this information in a meaningful way, in one waveform, what I compute is what is called a root mean square, RMS. So it's this red line over here. It's basically the instantaneous power across trials. It's a way of summarizing activity across these many trials into one time series. So this is the brain response to just a random sequence. So there's an onset here, there's an onset response, then there's a sustained response that continues throughout the sequence, and then there's an offset response. And you can see these little squiggles that are riding on top. These are responses to individual tones. So our tones are 50 milliseconds long. Now, if we produce a stimulus that has a transition from random to regular, it looks like this. So you see the responses overlap exactly, which as they should, up until, so this is where the transition is actually happening. The overlap continues for a while. In fact, this point here is 15 tones. So this is exactly the time that we observed in the behavioral experiment, and exactly the time that the ideal observer indicated as the point where you can start detecting the sequence. And at this point, we can see a divergence in brain responses between random and regular patterns. And in fact, in some conditions, we can see this even in individual subjects. So this is data from one participant, showing the same thing. And this is a single trials from that participant, showing that we can observe. So this is a very strong effect. And the bottom line with this experiment is that we can observe with MEG, we can basically observe the process of discovering a pattern with millisecond resolution. So we can see the brain discovering the pattern. And that the brain responses, and especially the latency of these brain responses in naive listeners that do not care, they're not focused on detecting the changes, mirrors behavioral performance. And this is the type of evidence that we use to conclude that these processes are automatic. So this is something that happens in your brain even when you're focused on other stuff. Your brain continually scans the environment, and whenever there's evidence for patterns, it would detect them, and in some cases bring them to your conscious awareness. Later on within that study, we realized that there's a simpler way to look at the process of regularity distraction, the regularity detection. And what I mean by simpler is more efficient. And that is that if we have a regular pattern and a random pattern, we can just look at their onsets. Because during the first cycle, the processing should be identical. During the first cycle, you don't know whether the regular pattern is regular or not. But later on, you should see brain responses that differentiate regular from random patterns. And that is more efficient because it means we can use shorter signals. It's also more efficient because in this kind of stimulus, at this point, there are two things that are changing. I mean, we introduce a regular pattern, so the brain detects regularity, but also we've introduced a physical change into the stimulus, right? The stimulus changed. And so it's possible that the brain responses that we see here are a mixture of these two things. Whereas when we use stimuli like this, we can see whatever is going to differentiate this signal from this signal is going to be purely driven by detecting patterns. And so this is what the data look like. So this is onset again, so this is 0. This is onset response and a response to the random pattern. And this is the response to the regular pattern. The divergence happens a bit faster now from onset, and this is the same is true for the ideal observer model. This is because when you have an absolute anchor, it's actually easier to find a pattern. And there are a few interesting points with this pattern that we can see. So the first one is yes, that the difference happens very quickly, and that it basically the difference between the regularity continues to build up to the second end of the second cycle, and then basically plateaus. So this suggests that at least with this way of looking at the data, there's no evidence that your brain accumulates or refines its representation of the pattern beyond the second cycle. Now, at this point, you can say, well, Maria, you told us that the brain is such an amazing statistical learning machine. Perhaps the brain just learned that you're using a pattern of 10 tones, because indeed, so far, we've only used patterns of 10 tones. So maybe I am overestimating how efficient the brain is, because if it knows that there's going to be a 10-tone pattern, it can do something slightly different to detect it faster. So in a different experiment, we now mixed up, so we presented patterns of 5 tones, 10 tones, 15 tones, or random patterns in random order. So in a given trial, you didn't know whether there's going to be a pattern, and if there was a pattern, you didn't know which one it's going to be. And these are the responses. So this is regular 5, regular 10, regular 15, random 20. They're all, the latencies are still, at least the latencies of 5 and 10 are like an ideal observer. The latency of the 15 tone lags a bit, the latency of an ideal observer. So what I'm showing here is this ideal observer detection data that I've shown you before, and this is overlaid on the brain data. And we can see that for regular 5 and regular 10, the detection happens at the same time as an ideal observer. For 15, so for patterns of 15, longer patterns, there's a delay of a few tones. So the brain responds a bit slower. This is interesting because it suggests that we can, using these kinds of paradigms, we have a way of estimating the capacity of the working memory or whatever memory buffer that is needed for these kinds of tasks. So this is not a standard working memory task because we're not asking anybody to remember anything. But to extract the pattern, some memory has to be used. And this gives us an idea of how much and under what conditions it fails. So back to this picture then. We can then, by looking by localizing brain activity at this point where the two patterns diverge, we can try to understand what systems in the brain are responsible for detecting irregularity. And when we do this, I must first say that this is magnetoencephalography, this is MEG. localization ability with MEG is somewhat limited. So I will show later some fMRI data. But based on the MEG data, we identify auditory cortex bilaterally and inferior frontal gyrus in the right hemisphere. And this network is commonly observed in MMN studies. So it's commonly implicated in change detection, in detecting changes or violations of patterns. And what is shown here is a similar network is also responsible for detecting the emergence of a pattern to begin with. And interestingly, we also see, and that's this point here, we also see activity in the hippocampus bilaterally. Now, the hippocampus is a brain structure that is highly implicated in memory and navigation. Its role in auditory memory is not very well investigated. So this is perhaps one of the first findings that implicate hippocampus in these kinds of automatic but memory-based paradigms. There's some work, there's a lot of emerging work in the visual modality that's implicating the hippocampus in statistical learning. And so this finding is consistent with those data. In fact, I have a few months ago, I've received a big grant in England to specifically study the role of the hippocampus in pattern detection. So we will know more in a few years. The most surprising aspect of these results, which might be surprising to, well, maybe not surprising to some of you, but surprising to those of you who have thought of sensitivity to patterns before, and that is the shape. So the fact that the difference between regular and random is manifested as increase in power that's associated with the regular patterns. And this is surprising because a lot of previous work has been showing the opposite. And logically, we might have expected the opposite. So if you can predict something, I mean, if you can predict the regular pattern, you can actually attenuate processing. There's no need to increase the energy that you are using up to code regular patterns. But in fact, I mean, we see this pattern, and I'll say a little bit later why I think we are seeing it and it hasn't been seen before. But we're seeing this pattern in different studies. So this is an fMRI study using similar stimuli, showing activity kind of broadly in auditory cortex. Yellow is regular and blue is random, and you can see that there's a larger extent of activation associated with regular patterns than random patterns. So that's consistent with the brain response, with the MEG brain response that we've shown. And similarly, you might remember these stimuli from the beginning of my talk. We see the same pattern with other signals that contain differences in regularity. So remember these scenes that I showed you? So we can have scenes where everything is temporally regular now versus scenes where they're temporally random. When we look at brain responses to the onsets of these things, so 0 here is scene onset, we can see that within 400 milliseconds, the brain response to these scenes varies such that responses to regular scenes are higher than responses to random scenes, just like we see with these spectral patterns. And we see it also in other stimuli that I'm not going to show you, where the patterns are not deterministic, so it's not a precisely repeating pattern, but where we change something about the statistics that makes something more predictable than something else. Our hypothesis is, okay, so why hasn't it been seen before? There's 3 reasons for why it might not have been seen before. The first one is that these responses are DC shifts, so they are very low frequency. And it's been a common practice in the field until recently to high-pass filter. So people removed these low shifts because within older technology, there was a lot of noise in those ranges. So people would just high-pass filter and then you wouldn't see anything. The other difference is that our stimuli are very rapid. So they're much more rapid than many of the signals that people use to study predictability. We chose to have them rapid for various reasons. First, because we want to model natural sounds, and natural sounds are rapid. Also, we want to eliminate conscious ability to track the signal. We didn't want people to listen and start predicting. We wanted to capture automatic processes, and that's important to do with fast signals. The third reason for why it might not have been seen before is that previous work has used very simple patterns. So patterns were usually just repeating tones. And the problem with these signals is that you confound repetition and hence adaptation, which results in a reduced response with predictability. Using these wideband signals that we are now using in the lab, we're able to create signals where you are not going to have much adaptation. And so likely we capture the process of the processes that are related to the predictability of the signal purely. And we have a hypothesis, and that's, and I will end with this, I have like 5 more minutes, that this sustained response indeed reflects a brain process that codes how predictable a signal is. And I will show you some evidence towards this. It's not conclusive evidence in the sense that we have now studies that are trying to capture this directly. But here is the evidence. So first of all, this effect does not depend on complexity. So if I showed you this, slide briefly before. So these are brain responses to regular 5, regular 10, and regular 15. They're very different in their complexity. So regular 5 is a simple pattern, regular 10 is a more complex pattern, and 15 is more complex. And they have different numbers of tones, so they differ in terms of adaptation, for example, but they all tend to the same value. In fact, 5 and 10 are overlapping. The green one, 15, is just below, but if you remember, that's also the signal that was where people performed worse than an ideal observer. So it's possible that what's coded here is predictability of a signal. Another way of looking at the same thing is looking at responses to random sequences. So what is shown here is data where we compared a response to the regular 5 condition. So that's the one that contains repetitions of patterns consisting of 5 tones. That's the red trace here. The gray trace is the same 5 tones, but presented in a random order. So it's not as predictable as the red trace, but it is more predictable than just the random sequence, because you know once you've been exposed to the pattern for a little bit, you learn that you can expect 1 of 5 tones. So this one's most predictable, this one's mid-predictable, this one's least predictable, and you can see the structure. We can also compare, so this is the same pairing for alphabet sizes of 10 tones and 15 tones. And interestingly, if you look at the responses to the random sequences, so this is a random sequence consisting of 5 tones, of 10 tones, of 15 tones, and this one is 20 tones. And you can see this gradation that's also associated with predictability, because a random sequence of 5 tones is more predictable than a random sequence of 10 tones, than a random sequence of 15 tones, and the random sequence of 20 tones is the least predictable. And the last piece of evidence is what happens when you violate a pattern. So I've been showing you so far responses to detecting patterns. And I mentioned that we also have stimuli where we're doing the opposite. So we have a pattern that's presented, and then we transition to a random pattern where basically the regular pattern is violated. So when we look at the brain response, it starts off looking, so this is the brain response to the regular part. You have an onset response, it goes up to a sustained response. Here we violate the signal. And what you do once you've violated it is you get a mismatch negativity response. So this is a response to the fact that there is a tone that you were not expecting. And then immediately thereafter, there's this down regulation in this sustained response that we interpret as the brain's decision, oh, this is now not predictable, and I'm going to downregulate the sustained response. The sustained response, I mean, if we take this evidence in favor of the sustained response reflecting predictability, it would make sense from a predictive coding perspective, in the sense that when you have something, when you discover something in the world, a certain channel that has predictable information, you will know that it's reliable information. This is information that you want to track. And so under this kind of explanation, you'd want to increase the gain on that channel. So, increase its salience, so to speak, in a way that then will enable you, if there is a violation in that channel, then you will know that there's a real change in the environment and you would be able to react really quickly. So this is our hypothesis for why the brain might be doing this. An alternative explanation, which I will just pose but not answer, is that perhaps this all reflects attention. So perhaps, like I said, regular patterns might be more salient. So what we might be seeing is this thing of a regular pattern capturing attention, and that might reflect the sustained responses. Indeed, I mean, this effect of discovering a regular pattern and this huge, I mean, this is 50% increase in power within the response, might reflect attention. It's harder to explain the downregulation, the very rapid downregulation in terms of attention, because it's not like if you violate a regular pattern, right after the violation, your brain says, oh, I'm not interested, and downregulates. So this bit is tricky to explain. But more generally, we've ran a series of experiments now, both behavioral and eye-tracking experiments that I could tell you about later if you're interested, that suggest with, I think, no doubt that it's not an effect of attention. What is it then? So it's likely not attention. It could be inhibition. So it could be that what we are calling an increase in power is actually an increase in inhibitory response. So when you have regular patterns and they're predictable, you might be actively inhibiting them, which we will see with fMRI and MEG as an increase in power, but it's actually kind of putting the brakes on something. It could be an additional source that becomes active when you discover regularity. Or it could be this hypothesis that I've just posed, that what this reflects is some kind of precision weighting. So it reflects some kind of internal, not translatable to attentional capture, but an internal salience weighting for predictable signals versus unpredictable signals. And that's basically the end of my talk, so I will just summarize what I've shown you. I hope I've convinced you that we can measure the accumulation of statistical information and detection of patterns in real time. And through these studies, reveal the fact that the brain, even the naive brain, continually and automatically scans the available sensory information or available auditory sensory information for the emergence of patterns and detects them very rapidly. And this is supported by a wide network of auditory cortical, but also frontal and hippocampal sources. And this is associated with this mysterious, huge increase in power. So regularity is associated by almost a 50% increase in the strength of the response. And we're trying to understand what that means. And there's lots of open questions, which relate to what does this DC shift mean? What is it that we are discovering? Is this process completely automatic? And of course, what patterns, what kinds of statistics can we discover? And we're working hard on all of these questions. Thank you very much for your attention. And this is the lab website.